This podcast is going to deal with cartilage, and cartilage can be considered a specialized skeletal connective tissue. Cartilage is composed of cells and an extensive extracellular matrix. The cells are relatively sparse. The major cell type, of course, is the chondrocyte. The extracellular matrix is very extensive. It amounts to about 95% of the total tissue volume. The extracellular matrix consists of various glycosaminoglycans, and collagen 2 is the major matrix protein, although collagen 6, collagen 9, collagen 10, and collagen 11 are also components of cartilage. Cartilage is a gel-like solid, but of course it's very pliable and compressible. Cartilage is avascular, and there are also no nerves or nerve endings in cartilage. So cartilage cells have to be maintained by diffusion, and cartilage has a relatively low metabolic rate. The three main types of cartilage that we will describe for the course are hyaline cartilage, elastic cartilage, and fibrocartilage. Major protein in hyaline cartilage, of course, is collagen type 2. Then there are proteoglycans and various multi-adhesive glycoproteins. Elastic cartilage, in addition to having the components of hyaline cartilage, is also very rich in elastic fibers. And then fibrocartilage has a higher amount of collagen type 1, but the matrix otherwise is similar to that of hyaline cartilage. Your learning objectives would like you to be able to recognize different types of cartilage on glass slides and on histological images, and you should be able to compare and contrast components of the extracellular matrix of different cartilage types with those of connective tissues in general. I'd like to spend most of the podcast talking about hyaline cartilage, and we'll use that as the model tissue for cartilage. Hyaline cartilage has a very homogeneous and amorphous matrix. The matrix appears glassy in the living state. You can see chondrocytes within lacunae, and that's evident on the diagram. You can see the chondrocytes within lacunae. Lacunae are just spaces within the cartilage matrix. In real life, the chondrocytes occupy most of the space of a lacunae, but oftentimes in a histological preparation you'll see the chondrocytes shrunken away. Now we also talk about isogenic groups. These would be clusters of chondrocytes within the cartilage matrix. So here is an isogenic group. These are isogenic groups. Composition of hyaline cartilage is mostly water, maybe 60 to 80% water. Collagen fibrils make up about 15% of the dry weight of hyaline cartilage. Of the collagen fibrils, collagen type 2 is the most abundant. These collagen fibrils are very small in diameter. They blend in with the ground substance. Unlike general connective tissue, the collagen fibrils don't aggregate into larger fibers. The ground substance in hyaline cartilage matrix is about 14% by dry weight, so about the same amount of ground substance as collagen fibrils. The ground substance consists of hyaluronic acid, various proteoglycans, and various glycoproteins. And then, of course, the cells make up just about the smallest component of hyaline cartilage, and the major cell type would of course be the chondrocytes. There are a few chondroblasts present, and there are chondroprogenitor cells. We might think of the chondroblasts as immature chondrocytes. This cartoon just gives an overview of the molecular structure of hyaline cartilage. So you can see chondrocytes like this in the cartoon, and these are collagen fibrils, and then hyaluronic acid, which is highlighted by these green molecules, aggregates with many different types of proteoglycan monomers, and those are interwoven into the network of collagen fibrils, as suggested. I'm not going to go over this cartoon here, but it's reminiscent of what we talked about in connective tissue as to how these proteoglycans assemble. So you can see these large hyaluronic acid molecules with proteoglycans that bind to them with various linker proteins. And then this slide just schematically suggests how the extracellular matrix interactions in hyaline cartilage might occur. So here would be, for example, some collagen 2 fibrils that are being bound and interacting with the hyaluronic acid, and the various glycoproteins that make up the large components of the extracellular matrix. On a histological view, hyaline cartilage has a dense connective tissue shown here, and this overlays a little bit less dense connective tissue called the perichondrium. Perichondrium is by definition a connective tissue associated with cartilage. Chondroprogenitor cells live in the perichondrium. Just below the perichondrium is an area of growing cartilage, and the cells in this area would be chondroblasts and immature chondrocytes. And the overall cartilage tissue grows here by appositional type of growth, and I'll talk about that in just a couple of minutes. The intense blue staining that you see here with a typical H&E stain is an area where there are mature chondrocytes, and the intense blue staining indicates synthetically active cells. Here would just be another view of the synthetically active chondrocytes stained with H&E stain. You can clearly see a lot of cytoplasmic basophilia and a Golgi halo indicating synthetically active cells. Note the Golgi halo at these black arrows. The electron micrograph confirms that these are synthetically active cells. There's a typical eccentrically placed nucleus common in metabolically active cells. There's lots of profiles of rough ER and Golgi. So again, the chondrocytes are very highly secretory in nature, and you remember from the discussions in connective tissue that the collagen precursors are secreted and are assembled into the collagen fibrils in the extracellular matrix. We can talk about three different matrix types in hyaline cartilage. We can talk about a pericellular or capsular matrix, a territorial matrix, and an interterritorial matrix. The capsular or pericellular matrix is a ring of densely staining material right around the individual chondrocytes. It primarily consists of collagen type 4, and capsular matrix has the highest concentration of proteoglycans. The territorial matrix, as shown on this cartoon, surrounds isogenous groups of cartilage cells, consists primarily of collagen type 2 and collagen type 9. It has a lower concentration of proteoglycans as compared to the pericellular or capsular matrix. And then finally, the interterritorial matrix surrounds the territorial matrix. This occupies the space between groups of cells. It has the lowest concentration of proteoglycans, and interestingly, proteoglycans are lost as cartilage ages, and this loss of proteoglycans occurs primarily in the interterritorial matrix. This cartoon is just to remind me to tell you about cartilage growth, and we can have appositional growth where there's additional cartilage being laid down at the periphery of a plate of cartilage. That would be appositional growth, as opposed to interstitial growth where the cells within the cartilage plate divide and the plate grows that way by interstitial growth. A couple of general notes I want to highlight for you. In adults, damaged cartilage is not repaired. Cartilage can take a lot of stress before it is damaged, however, but once it is damaged, it generally cannot repair itself, and that may be because it's avascular. Just another point, hyaline cartilage of the articular joints does not contain a perichondrium, and hyaline cartilage, as you know, provides a model for skeletal development in the fetus by a process called endochondral ossification. And we'll talk about endochondral ossification later in the course. These are just some histological images of hyaline cartilage, just a lower power view on the left, and you can see the nice staining components of the matrix. So you've got territorial matrix staining darker, and then the interterritorial matrix, just a higher power histological view. You can see capsular matrix right around the isogenous groups. You can see lacunae, and you can see chondrocytes in the lacunae. And in this image, of course, the chondrocytes are shrunken away. Here is just an electron micrograph of hyaline cartilage showing some chondrocytes, and note the association with these collagen fibrils. like to just make some general comments about hyaline cartilage, elastic cartilage, and now fibrocartilage. This cartoon just shows the relative distribution of the cartilage types in the body. Hyaline cartilage, of course, resists compression. It provides structural support, so it's found in the fetal skeleton. It's found in epiphyseal plates. It's on the articular surfaces of synovial joints. It's found in the costal cartilages of the rib cage. It's in the nasal area, so there's nasal cartilages, and of course, hyaline cartilage is found in the larynx, and then in the trachea and in the bronchi. Hyaline cartilage can calcify with aging, and the principal cell types, of course, are the chondrocytes and the chondroblasts. Elastic cartilage is more flexible, so you find it in the external ear, in the external auditory meatus, in the eustachian tube, and there are cartilages in the larynx that are also elastic cartilage. Again, principal cell types are the chondrocytes and chondroblasts, and elastic fibers are very prominent in the matrix of elastic cartilage. Otherwise, the matrix of elastic cartilage is very similar to that of the matrix in hyaline cartilage. And finally, fibrocartilage resists deformation under stress, so you'll find it in the intervertebral discs, in the articular discs, so like in the sternoclavicular regions and in the temporomandibular joints. You find it in the menisci of the knees, and you find it in the wrists and ankles. An interesting point, there is no perichondrium found in fibrocartilage. Fibrocartilage, like hyaline cartilage, can calcify with the aging process. The principal cell types are the chondrocytes and fibroblasts. One doesn't really identify chondroblasts in fibrocartilage, so the immature cells are fibroblasts, and perhaps the fibroblasts can differentiate directly into the chondrocytes. This is just a general histological view of elastic cartilage. You can see the intense staining, the fibrous nature of the matrix. It's not as glassy as that of hyaline cartilage. Here are two images of elastic cartilage with a specialized silver stain to highlight the elastic fibers. So you can see the fibrous nature of elastic cartilage. You can play a trick with your condenser diaphragm on the light microscope. That is, if you stop down the condenser diaphragm, elastic cartilage will begin to look like it's got this network of fibers in the matrix. If you do that on hyaline cartilage, you just get a darker image, but you don't see that fibrillar type network. And then this is fibrocartilage, both at a lower power and at a higher power. You can see the isogenous groups in the fibrocartilage. I want to just conclude the podcast with a couple of clinical correlations related to cartilage. We talk about osteoarthritis and chondrosarcomas. Osteoarthritis is a degenerative joint disease. The pathogenesis of osteoarthritis is not really understood, but it's involved with aging or injury to articular cartilages. It usually becomes manifest in the 6th decade of life with chronic joint pain and joint deformity, especially on the weight-bearing joints. There's decreased proteoglycan production, and as a result, there's less intracellular water in the matrix. The chondrocytes produce interleukin-1 and tumor necrosis factor alpha. That means there's less matrix proteins. There's eventual spread to bones as the articular cartilage is lost, and there's a progressive reduction in someone's mobility. The treatment for osteoarthritis focuses primarily on symptom relief and pain management. Now, with respect to chondrosarcomas, these are malignant tumors of cartilage. They're slow growing, but they can account for somewhere about 3 to 4% of primary bone tumors that are diagnosed in the United States. Matrix components are overly secreted. Chondrosarcomas are more frequent in men, and they begin to be seen by the 4th decade of life. They originate in the axial skeleton. There is a tumor grading system for chondrosarcomas, just like there are for many types of cancers. The higher the grade, the poorer the prognosis. Histologic clues for chondrosarcomas include binucleated or pleomorphic nuclei within single lacunae. Interestingly, SOX9 expression is important in tumorigenesis of chondrosarcomas, and treatment of chondrosarcoma focuses primarily on surgical intervention.